Hello everybody. This is the finishing up of the class we were doing on Tuesday. I just wanted to record this and get this in the book so we can move on next week. You have your online class available as of now, I hope. Hopefully the My Courses problem has been solved. So I just wanted to go over the last few slides on conformational changes of proteins and why they are important. So, as I said last time, conformational changes in proteins happen when the protein, whether it be an enzyme or a structural protein or whatever, when that protein binds something. And that something could be substrate, DNA, some kind of small molecule that can act as a regulator or whatever. And they often will occur, these changes. They often are, the result of them is part of regulation of enzyme activity. And so here's a classic example here that I began to talk about on Tuesday, the binding of 2,3-diphosphoglycerate to the oxygen oxygen binding to hemoglobin. So you can see in this diagram here we have oxygen binding to, this is one subunit of hemoglobin, a small part of that subunit, where we have this thing called heme that is present in all hemoglobin, and it has a certain shape. They're calling it here a dome shape, but when oxygen binds, it undergoes not only does the cofactor undergo a conformational change, but also the protein itself undergoes a bit of conformational change. And so you can see that here when it's oxygenated, the heme looks different and the protein looks a bit different. But how does 2,3-diphosphoglycerate affect that? And so again, stepping back from hemoglobin, a mature hemoglobin A molecule is a tetramer. So that means it's 4 subunits. And you can see here in this drawing, there's 4 subunits. Each are colored in a different color here, and this is vaguely what the proteins look like. Each of the peptide chains looks like when they are complexed all together. So there's 4 subunits. 2 are called alpha chains. You can see the alpha chains are labeled right down here, and 2 are called beta chains. The beta chains are colored more brownish, and they're labeled up here. And there are 4 molecules of, again, I said a cofactor, this heme. And here's the chemical structure of heme, and it looks pretty complex here. There are 4 molecules of this heme bound in as cofactors for hemoglobin. And that's where the oxygen binds. So they each have this iron center right here, as you can see, and that's denoted by the red dot in that drawing. And oxygen is bound to hemoglobin by the assistance of these heme molecules. And so that becomes important when we consider the activity and the regulation of oxygen binding here. So what is diphosphoglycerate doing? Hemoglobin A will bind oxygen much more tightly in the absence of this diphosphoglycerate, or we'll call it DPG. So if there's no DPG around, hemoglobin will bind oxygen much more tightly. And DPG itself, and you can see here's the chemical structure. It's got 2 phosphate groups. It has a net negative charge. So it's a charge of -3, which tells us a little bit about what kind of intermolecular forces are at play here, what kind of interactions this molecule can make with the hemoglobin protein. And so where this molecule binds is kind of in the middle of this 4 subunit protein complex, and it complexes with a couple of different amino acids, 2 histidines that have a positive charge in their side chains, and the negative charge of the phosphates can complex in weakish bonds, salt bridge type bonds, in complex with these histidines, and a lysine, which is positively charged and will interact again somewhat weakly with the acidic moiety here of the DPG. So this allows for the negativity allows for DPG to bind to positively charged amino acids like this. And there's these 3 amino acids, 2 are histidine and 1's a lysine, at the N-terminus of the beta chain of the hemoglobin. And that N-terminus is obviously located somewhere near the center of the complex, because that's where DPG binds. So what does this binding mean to us? So if we look at the affinity of deoxyhemoglobin to DPG, it's about 8.5 times 10 to the 4th per molar, is what that should say, not per meter, per molar. And fully oxygenated hemoglobin has an affinity for DPG that's much, much less, 330 per molar. Okay? So that tells that there's some sort of competition, not for the oxygen binding site necessarily, but there's some sort of competition in binding the hemoglobin between oxygen and this DPG. And the binding of oxygen to deoxyhemoglobin requires removal of this DPG. Oxygen and DPG therefore compete for binding to hemoglobin, although they're not binding at the same site, as you can see here in this diagram here, where we have oxygen bound to the hemoglobin, each of the subunits, so 4 oxygen molecules bound to each of the 4 subunits at the iron of that heme, and then we have DPG bound here in the deoxyhemoglobin, and that is precluding or kicking out oxygen, because the conformation is such that oxygen is able to bind much less, with much less frequency, in hemoglobin that has DPG complex with it. And that happens for a few reasons. It can be, the increase of the DPG concentration. It could be a falling partial pressure of oxygen. It could be an increasing partial pressure of carbon dioxide that is causing this. And there are reasons why hemoglobin would be you'd want it to be less specific to binding oxygen under situations like that. So basically DPG is acting as what we call an allosteric regulator, and it's binding the hemoglobin, and it is to a certain extent precluding the binding of oxygen. And you can see this in a saturation curve if we look at oxygen saturation of hemoglobin, or oxygen binding of hemoglobin, with and without DPG. So the green line here is without DPG, or the BPG, or DPG. This is bisphosphoglycerate, diphosphoglycerate, same thing. But this is without, and you can see that at low oxygen concentrations, even at low oxygen concentrations, the saturation happens very quickly of deoxyhemoglobin in the absence of this diphosphoglycerate. But with diphosphoglycerate, we can see that the saturation will happen a lot later. You need a lot higher partial pressure of oxygen if you want to get full saturation of hemoglobin in the presence of this bisphosphoglycerate or diphosphoglycerate molecule. And there is a physiological reason for that, because hemoglobin is ultimately delivering oxygen to tissues. So once it is bound, we want it at some point to become unbound. And so this kind of smooths things out a little bit and allows that binding and unbinding to happen more readily. All right. Finally, let's take a little foray into the structure of biological membranes. We've talked about them a very little bit here. I'll just explain the arrangement of membranes briefly. So membranes obviously cover all living cells, either with or without cell walls. And what they are is they're a bimolecular, what we call leaf, and you hear membranes talked about as if they're leaflets, and that's where that comes from. So it's like a leaf arrangement of phospholipids. And some in humans, they contain cholesterol, and in most organisms, there's some proteins floating around in there. And the lipids are oriented in such, and you can see in this diagram here kind of, that they have these hydrophobic tails and they're in contact with each other. So they're, again, it's these hydrophobic interactions that are causing the membranes to assemble like this. Just like hydrophobic interactions play their role in protein assembly, we're seeing this in the plasma membrane also. That this is one of the things that's driving the assembly. And these hydrophilic heads, the phospholipids, the phosphate, glycerol, and perhaps some other things, the polar heads are on the surface. They're on either surface of the membrane bilayer because they are facing tails in. So you've got polar heads on one surface and you've got polar heads on the other surface, thus making the membrane leaflet. And these membranes are held together by non-covalent interactions pretty much exclusively. Proteins and lipids can diffuse laterally, they can rotate, they can diffuse laterally. They don't do so much of this transversal diffusion without protein help, but they can certainly rotate around where they are. They can laterally diffuse to a new place, and proteins, this shows a lipid doing that, proteins can do that as well. So they can diffuse laterally through the membrane. So and then we say the membrane has fluidity. How much fluidity it has depends on the phospholipids that are in it. So if we've got unsaturated hydrocarbons in your phospholipid, they're going to have these kinked tails here, which will create some space in here and allow for more lateral diffusion, allow for more movement in that membrane. If you've got very straight saturated hydrocarbon tails in your lipids, you're going to have a very tightly packed structure here, and you can see that here, and that makes it more viscous and harder for lipids, for individual lipids, or for proteins to move around laterally in a membrane. Usually there's some combination of the two things in most biological membranes. So the flexibility of the hydrocarbon tails dictates how fluid the membranes are. And cholesterol, here's a drawing of the structure of cholesterol right here, is this ringed structure, very hydrophobic structure that can get packed in, and we see this in this diagram, cholesterol packed in between the phospholipids, and that can reduce the coiling of hydrocarbon chains in the lipids, and cholesterol ultimately reduces fluidity in lipid bilayers, and that makes our cells, mammalian cells, makes them a little more sturdy, because we obviously don't have cell walls around our cells. So cholesterol toughens up our cells' plasma membranes. The shapes of lipids can play a role as well, and they do play a role in the formation and the shapes of ultimately the membranes that they're in. And so membranes tend to tightly pack their lipid components, and that's to exclude as much water as possible in the hydrophobic region, because we don't want them to be super leaky in our cells. That could cause some real problems. So some phospholipids do have shapes that may deviate from, the straight cylinder-like lipids of straight-chained hydrocarbons. And so if you've got straight-chained hydrocarbon tails, you might have this cylinder-like lipid and have this flat, well-packed lipid membrane. If you have some more inverted, some kinked tails, hydrocarbon tails, you might have a more triangular looking or more rhomboid looking lipid shape, and that allows for some curvature, this negative curvature here that you can see. and if you have real sharp, even single side chain, hydrophobic side chain here, kind of like a detergent, you can have this really tight curvature here for these lipids. And this happens a lot with detergents, and if you have lipids that have only one, if you have monoacyl glycerols and things like that as your lipid backbones, then you would see this tight curvature here. So different conformations can be introduced depending on the shape of the lipid, and that's dictated by the chemical structure here, whether you have double bonds, single bonds, how many chains you have. Those are the things that need to be considered here. And we can quantitate lipids in certain ways, and that's how they interact in solution, both with each other and with the water that surrounds them. And there's a concept called critical micelle concentration. This is true for lipids and detergents here. And roughly defined, critical micelle concentration means the lipid monomer concentration at which appreciable amounts, which seems to be greater than 5%, of micellar aggregates. And what's a micellar aggregate? Well, here's one right here, where they form this kind of ring that kind of looks like you see soap bubbles or globules of soap floating around in solution, or globules of oil floating around in an aqueous solution. Those are micelles. Where you have the hydrophobic tails interacting with each other, and in solution you have the hydrophilic heads, the polar heads, interacting with the water that surrounds them. So this is essentially the concentration to which you start to see these micelles in the medium, and they appear to be in equilibrium with the solution here. And so this micelle is the smallest and most thermodynamically feasible shape that these polar, nonpolar, aka amphipathic compounds can make in an aqueous solution here. And the longer the hydrophobic tails of the lipid, the smaller the critical micelle concentration will be. And presumably that has to do with the ability and the amount needed to successfully interact and geometrically make a micelle here. So for example, if we look at phosphatidylcholine, if we have lipid side chains, hydrophobic chains that are only 5 carbons long, you need about 90 millimolar phosphatidylcholine 5O to make a micelle. But if you have carbon chains that are 14 carbons long in phosphatidylcholine, you only need 6 nanomolar of phosphatidylcholine 14O to make to hit that critical micelle concentration. So this is one way we can quantitate lipids in solution. Okay, so what we learned between me talking right now and Tuesday's lecture, we have osmotic pressure, which is a colligative property of biomacromolecules that we care about. And we can measure that a couple of ways: sedimentation, equilibrium. And we can measure that via light scattering. Electromagnetic forces play a role in the intermolecular interactions of biomolecules. Those are the forces we pay most attention to. There are different types of bonds, and these bonds have different energies. Van der Waals forces have the lowest energy. They're more just, by force of proximity. Covalent bonds are some of the highest energies, and we know this because of the energies it takes to break most covalent bonds. The concept of solvation is helpful to fold proteins properly in the cytoplasm and in solution. This ensures proper interactions among amino acid side chains, which in turn ensures proper folding of proteins. Protein conformations are often used for regulation of cellular activities, and there's a lot of literature on conformational changes in proteins leading to some kind of regulation, whether it be regulation of gene expression or regulation of activities of enzymes. Not just the hemoglobin, although hemoglobin is a pretty good example of that. Membranes will form a bilayer structure, and again, this is because of the nature of their chemical makeup, and B, their geometry. That has the hydrophobic tails in the center interacting with each other, hydrophilic heads at the surface, and those are interacting with water in aqueous solution. And again, the lipids in question here are what we call amphipathic, which means they have a hydrophobic section and a hydrophilic section. Okay, so that is the rest of it. Your online class should be up on my courses and available. If you have questions on that, please don't hesitate to either come to my office or send me an email, let me know. I will do my best to help you. And we will meet on next Tuesday, to discuss biothermodynamics. Until then, have a good weekend.